So thank you for coming. I'm Richard Fox with Inscripta, and I want to start with a little bit of a background on myself, because I think it will be important to sort of understand a lot of what we're trying to share with you folks today. So I'm actually a nuclear engineer by training, more years ago than I care to say. But I was lucky enough to learn about the power of evolution and evolutionary biology over 2 decades ago. And by twists of fate, I ended up working in the biotechnology industry for some really interesting companies developing exciting technologies, especially around the field of protein engineering through methods like directed evolution. And so that was a really exciting time, and we had a lot of fun working in that field. The excitement and passion around evolving or engineering proteins for different functions ultimately left us wanting to be able to apply the kinds of strategies and tools we had developed in that sort of small-scale biological optimization problem to larger sequences, namely pathways and genomes. But the tools were inadequate at the time, so we had to wait a few years. But fortunately, a couple of years ago, I was lucky enough to talk to some folks who were developing a CRISPR-based genome editing technology company and platform. And that company is Inscripta. So that's what I'm going to be talking about a lot today and sharing where we're at. But I want to start with sort of a background in terms of the challenges associated with genome engineering, and then get to the Inscripta solution, and then share with you, we think, a motivating example with a demonstrated application of how the technology can be applied. And then finish with what's next for Inscripta and the rest of the world as we come forward with a platform that I'll describe at the end of the presentation. Okay, so in terms of the challenges associated with biological engineering, it turns out it's really difficult. So there had been some laudable aspirations about 10 or 15 years ago to sort of recast engineering biology as done in the same fashion as mechanical or electrical engineering. So if you could sort of characterize component parts and put them together in predictable ways, you'd be able to achieve whatever outcomes you're interested in. And so while that was a wonderful aspiration, it turns out it's met with mixed success. And it's because we can't really reduce to practice building biological systems in the same way you build, say, transistors or integrated circuits. So for the foreseeable future, we're going to have to take very empirical approaches to letting nature tell us what works and what doesn't when it comes to optimizing for performance in complex biological systems. So it turns out that the strategies and tools for doing this is actually very well developed in the field of enzyme and other types of proteins engineering. And a couple of key components to that successful strategy includes the ability to generate lots of diversity and then optimize over that diversity, that very large combinatorial space. So what is diversity generation? So if you look at any sequence, whether it's a protein or a pathway or a genome, it's hard to know where to intervene to try to obtain some increase in function or performance that you're interested in. So a typical protein, 300 residues or so, 20 amino acids at every position, that's already many thousands of possible changes you could make. Which ones do you make? And then the problem just gets bigger as you go to pathways and genomes. So that's a central challenge that's particularly difficult with the larger sequences that you have. And then even after you establish some mechanism or strategy to be able to identify what's important in your sequence, then you're left with a very daunting combinatorial optimization problem where you have to search over various combinations in order to find improved sequences. All right? So we'll share with you some examples to give you some insight into how this works at the gene scale. But where we really want to go is how do we take this kind of strategy to work at the pathway and genome scale? So what's missing? Because in principle, we had the playbook for many years, but we didn't have the tools. And so the big piece that was missing in the tool set was the ability to do genome scale writing. Okay? And that will be the central emphasis, especially as we get later in the presentation. Okay, so I've already kind of alluded to this a little bit. So if you look at sequence space, whether it's a protein or a pathway or a genome, the number of possible combinations is exceedingly large. So for a protein with 20 amino acids at every position, several hundred residues, you multiply out all the possible combinations and the number is astronomical. And the problem just becomes more difficult as you go to pathways and genomes. So how do we navigate this space? It turns out we can take great inspiration from evolution. It is essentially our guide to navigating these large sequence spaces. So evolution's been around, of course, for billions of years, practiced by nature in various forms. But it's also been well applied to plant and animal domestication over hundreds and thousands of years. And I think most folks are aware of the fact that Francis Arnold won the Nobel Prize in Chemistry for directed evolution carried out at the molecular level. So we can take great inspiration from evolution to try to navigate these large spaces. And it's sort of worth an aside here that in addition to all this natural evolutionary process, one of my first exposures to evolutionary principles was in the form of something called genetic algorithms. These are actually been applied over decades in the computer science community, and they basically take the principles of evolution, reduce them to in silico processes, and they're able to search through large combinatorial spaces for completely unrelated optimization problems. And so when I got into this field about 20 years ago, that being my first experience, it was really fun to see this kind of come full circle back into the wet lab, as it were, those evolutionary processes. So let's just talk a little bit about two primary forms of evolution. One is an asexual process, the other is a sexual one. So let's distinguish those two. So in an asexual process, you start with some parent sequence, whether it's a gene or a pathway or a genome, whole organism, and it undergoes some diversity generation. So this, where do you mutate to make changes? It could either be random or directed. Typically, in an asexual process, the best variant wins out overall, and then you use it as a starting point for your next set of mutagenesis experiments. And then you repeat that process as many times as you need to get to where you're going. Asexual evolution, very powerful. It predated sexual evolution in nature, still is around today. But there's another format of evolution that in many ways is much more powerful and efficient, and that's the sexual form. So if you take a starting parental sequence and you diversify it, instead of taking just the best variant, you take all of the genetic material in the winners, and then you recombine that through a recombination process. And there's lots of different ways to do this. The gentleman on the right, his name is Pim Stemmer. He invented this really super cool technology called DNA shuffling a number of years ago in the mid-90s. And the idea was to basically randomly fragment genes and let them recombine, related genes, and let them recombine in random ways to create sort of this hypersexual population of progeny that could then be selected for improved traits or phenotypes of interest. That core technology, DNA shuffling, led to the formation of a number of companies, one of which, or a couple of which I was associated with a number of years ago. So that process works really well at the gene level. Here there's a study that was reported actually some time ago, but I think stands the test of time and I think is a great demonstration of how powerful this recursive process is. And it was done on a microorganism that produced an industrial relevant antibiotic called tylosin. Okay, so there was an industrial partner that these folks worked with that had evolved using this classical asexual approach over many years, 20 years, and 1 million assays to get to their final endpoint. And these are different formats for introducing random mutations in your genome. So that process does work, but it took a lot of effort and time to get there. So instead of that, what my former colleagues were able to show is that doing the initial diversification round to collect those individually beneficial SNPs in the genome likely, and this is well before there was genome sequencing and other technologies to read back out what the changes were, you could blindly recombine the different parents and get much larger improvements in function for very limited resource allocations. So in particular, what they did is they screened a couple 10 or 20,000 colonies, or isolates I should say, in the first round to generate that diversity, and then they rapidly recombined it and followed up this fitness trajectory much faster. So with just another 1,000 isolates screened in 2 rounds following, they were able to achieve the same endpoints that took 20 years and 1 million assays. Here it was only 1 year and 24,000 assays. So I think this demonstrates well the extreme power of a recombination technique, even at the genome scale. So you might say, well, the problem is solved, but it's not for a lot of reasons. 2 of them is that this sort of mating process is only amenable to certain organisms, so it's not widely applicable, and also the diversity generation strategy here was just random mutagenesis, and that turns out to be a very inefficient way to generate diversity in many cases. So we're very interested in exploiting this, but in a more powerful way with the technologies I'm going to share with you here in a moment. So again, we're seeing this idea that there's diversity generation followed by combinatorial optimization that works very well for optimizing large sequences. So before we get into more about how this combinatorial search process works, it's worth pausing a little bit and talking about your diversification strategy. Where do you find these individually beneficial edits or mutations? And you can think about one strategy, which is to say, as a scientist, we're very happy and satisfied when we can predict what to do and the experiments turn out in our favor. So if you have a good ability to generate ideas that have a high probability of winning out and being successful, but you only test a small number of those things, then when you multiply these 2 things together at the end of the day, after that exercise, you may only have 10 hits, okay? And 10 individual mutations, while good, represents a small amount of diversity that you can carry forward over a number of rounds of evolution. So instead of pursuing this strategy, or I should say in addition to pursuing this strategy, we certainly are very excited about any rational ideas that people have. There's this ability or thought to take a lower success rate, but in favor of looking at many more variants screened. So when you multiply, say, a 1% success rate with 10,000 variants screened, you now end up with 100 individual hits. And this represents a lot more evolutionary fuel, so to speak, for the optimization process that we'll spend a little bit of time talking about. Okay, so I think this is sort of best summarized by Voltaire's quote, "The best is the enemy of the good." So the idea, so a sports analogy would be, if you're, don't pretend to play golf and lining up your best perfect shot when the goal is soccer or something that takes a lot of shots on goal to get the wins in. Okay? So that's just a little bit about diversity generation. We'll spend a fair bit of time showing examples of how we can go much broader than just targeting a few edits in a genome, in a pathway or a genome. So now once you have a bunch of diversity and you're sitting on a large pile that you can then start to recombine through various technologies, question is, what does that distribution look like, and what are some of the key patterns or aspects that you need to understand to be efficient about searching through that space? So it turns out if you make these combinatorial libraries and they're of any sufficient complexity, then as you start to fish out or screen through that pool of variation, then when you look at the number of things you've evaluated here on the x-axis versus the number or the best fitness that you've pulled out of the population at any given screening effort, then it turns out there's this really interesting property that for almost all non-trivial combinatorial libraries, you almost always get a curve that has this kind of shape. In particular, after you look at the first few hundred or first few thousand things, it's very hard to find something better than anything you've already seen. There is some additional improvement, but you have to screen much more deeply into the population to see some real material gains. The y-axis itself and the absolute numbers is not so important, it's just a simulation, but we have empirically demonstrated these kinds of curves and this sort of relationship over many real protein engineering projects. So you might be tempted to think there's nothing better in the population, but if we switch from a linear scale over to a log scale, it turns out there is much better things in the population. It's just really, really hard to find them. It's like getting struck by lightning or winning the lotto. So you can continue to sort of screen, deeply, in the teeth of these stupefying odds, but you'll never find something out here. It just would be incredibly unlikely. So instead of doing that brute force evaluation of the whole space, let's pursue the strategy that we just talked about earlier, which is to say, well, after screening, say, just 1,000 things, take your best parents, just like what nature does, and recombine it so that you then end up on a much more favorable trajectory in terms of screening versus your fitness that you've gained. So you can see just in the second round, you get a much better return on investment. For the next 1,000 things you screen, you end up at a much higher fitness than anything you got from deep screening. And you just repeat that process over several rounds, and you end up really kind of interesting or shockingly, if you've never really paused to consider it or seen examples of this, a fitness level after looking at only 4,000 candidate solutions that would have required 100 billion evaluations from the original population, okay? So I hope this gives you an understanding of why sex is so great. If there's ever any wondering about why we invest time in mate selection and courtship displays, nature hit upon sex because of this ability to navigate these really large spaces, all right? So that's great. This approach and the technologies that implemented it in the wet lab have been around now for quite some time. On about 10, 15 plus years ago, as we were working in this field, we began to realize that there was an opportunity to take the genotype and phenotype information that was being generated during the evolutionary process and mine it to try to guide the search and accelerate towards higher fitness more rapidly. And I'll show you just an example of that on the next slide. But I do want to sort of on this slide leave you with, I think, my favorite quote in all of science from the legendary R.A. Fisher, which is natural selection is a mechanism for generating an exceedingly high degree of improbability. And I think you understand now where that comes from. Okay, so the machine learning guided approach is one where you mine that genotype and phenotype space and you get more rapid jumps in the fitness. So let's talk about how this works. So sticking with this notion of improbability, Richard Dawkins, the prolific evolutionary biologist and author, wrote this wonderful book called Climbing Mount Improbable, okay? So the improbability part is, I think, well understood now that we talked about the last slide. It's very hard to find randomly just really good solutions. The other part to this, Mount Improbable, is an idea that's been around for quite some time now. The American geneticist C. Wall Wright had this concept that you can think of the evolutionary process as a high-dimensional walk over a fitness landscape where latitude and longitude is genotype and elevation is your phenotype. And the idea behind a machine learning or statistically guided approach to evolution is to say, let's make a local map of genotype to phenotype relationships and then use that map to decide where to go next. And the implementation in silico can be as simple or as complex as you want, especially depending on how much data you have. But the idea is actually quite straightforward, which is to say you want to build an in silico map of this sequence function space that takes as an argument a given sequence and tries to predict activity. And these are the days of machine learning and artificial intelligence, and depending on how much data you have, you can build quite good maps of your system. So that's the essential idea. It's been very well demonstrated to work exceedingly well in the enzyme and protein engineering space, and it's a vision for us to apply to pathways and genomes. Okay. So let me sort of bring it all together and show you an example that is done at the enzyme scale in some work that I was fortunate to be involved with a number of years ago. So here we were trying to optimize an enzyme for a given property. In this case, it was thermostability, but it's generalizable, the process, for pretty much any trait or phenotype you're interested in. And the idea was to say, okay, we want to improve this enzyme over some number of rounds of evolution. So let's start with the diversity generation first, which I talked about, right? Let's take a lot of shots on goal. So if if you've ever looked at a protein and multiplied out all the single amino acid mutations you can make, 20 times the length of the protein, you end up with library sizes typically in the mid-thousands, okay? You can create those libraries. There's very cost-effective, fast methods to do this, and then you can start screening them through various, typically high-throughput screening methods. As you sample from that pool, a key point to realize is that you don't need to see everything in the population. And because you're sampling from a pool, to guarantee that you see everything would require you to massively oversample this population. But it turns out that you can sample typically anywhere from 30 to 50% of the theoretical size and get to lots of beneficial diversity. So let me actually show you how that works, or how that worked in this project. So after screening these individual single amino acid mutants, we ended up with something that was 70-fold improved after just that first round. But more importantly, we had 84 distinct beneficial mutations in our protein that we could then use in this recombination process that I've already described a little bit. So let's go ahead and do that. And what we did is we designed libraries that are actually quite large now relative to the diversity generation, but we screened less from that population, both in absolute terms and in particular on a relative basis. So here we're looking at less than 2% of the theoretical library, but as I think I've been able to show you on the last slide, or the last two slides, that you can navigate this space very efficiently by sampling a small fraction, but here we were able to get another order of magnitude jump in phenotype, and there's still a lot to be gained from this diversity that you're optimizing over. So then we take that machine learning guided approach to build these genotype-phenotype maps to drive the evolutionary process forward over a number of rounds of evolution. Okay? It's a very efficient process. It's pretty much, this represents the template for the state-of-the-art methods in enzyme and protein engineering. And just to give you kind of a time scale, when we did this kind of project a number of years ago, it would take 4 to 6 weeks or so if you have all the infrastructure in place. So you can move quite fast. The best practitioners of this can move even faster today. All right? So it can't be overstated that high coverage is not necessary to achieve successful outcomes. You do not need to brute force your search of this space. Nature's been able to do it for many years quite effectively, so we're inspired by that. Okay, so that's a lot about protein engineering, but as I kind of mentioned in the opening, we're really using that as our guide for what we want to do at Pathways and Genomes. So in protein engineering, you're typically, now as we switch to pathway and genome engineering, I want to contrast what you typically do in protein engineering, which is usually making small edits, usually at the codon, the amino acid change. And that's fine, that can be done, but to move to the pathway in the genome, in addition to doing sort of amino acid mutagenesis on proteins, you also want to be able to target, say, ribosome binding sites, or promoters, or terminators, or combinations of all these different edit types. So you really need to have a high variety of edit types at your disposal to really go and intervene in fairly unrestricted ways in your biological sequence. But that's not enough. You also need to be able to do this at the genome scale. You need to be able to go everywhere in the genome. This is a Circos plot of the E. coli genome from an application that I'll show you in a few slides. It's just demonstrating the need to be able to go genome-wide. Okay, so where is the field at today? It looks like, for now, the world is sort of trapped in a couple of less than favorable quadrants. And the representation here is given the number of genomic locations on the x-axis. These are disparate locations in the genome. And then the variety of edit types, which I just talked about on the last slide, is this, are you making single base pair changes or just knocking something out, or are you making larger changes in your sequence? And it turns out for a lot of applications like protein engineering or pathway engineering, where you're just hitting a few targets in the genome, you can carry this out relatively easily with some of today's technology, but it's very hard to go to a lot of genomic loci and get a lot of variety of edit types as well as high numbers of edits in that system. On the flip side, the other axis that people are sort of trapped, or quadrant that people are trapped in today, is one where you can target the whole genome, but you're quite limited in terms of the edit types. So for example, genome-wide base editing, which is sort of a new technology that's very exciting, is great, but it's limited to sort of single base pair changes. And then CRISPR is basically dominated for the most part by knockouts, right? People call it editing, and it is editing, but it's a very limited form of editing, right? It's as if I gave you a word processor and said, here, you can make single letter changes, or you can knock out a paragraph or a sentence or two. If that's the only thing you had, you would take it. But what you really want is a word processor that can make larger changes in very directed ways all throughout the document. So taking that analogy to genome engineering, where we need to be is to have the ability to have a high variety of edit types as well as high number of genomic loci, this is going to sort of unlock the potential of this field of genome engineering for industrial applications, as well as a lot of applications in sort of maybe what you might think of as basic science, just discovering how nature works. There's still much we don't know about genomes, despite all the years that we've studied even simple organisms like E. coli. So in order to get to here, we believe that effective solutions are going to require, first, unprecedented scale, which is already this sort of notion I've talked about here, which is high variety of edit types and large number of genomic locations. But that's not sufficient. In addition to that, we also need to have superior performance in the form of high editing rates. So it turns out a lot of existing technologies, when you actually run through and look at what is the editing performance in terms of how many cells are actually edited in your population, it can be quite low, too low for many practical applications. So you need to be able to confer high editing rates. It's a very key aspect for success. And then finally, greater access. So by that we mean at least two things. One is the ability to carry out this process in a fast, cost-effective, reliable, essentially turnkey or automated format, to really relieve researchers the burden of having to struggle with the technology to make their edits, and rather focus their time and energy and resources on the science or the questions that they have. So that will figure greatly in what I'll lay out over the latter half of the presentation. And then the other piece to greater access that's particularly important in a lot of applications is to have the FTO around the things that you make, right? Having the IP freedom to own your inventions. And for anyone who's sort of followed CRISPR, you'll know that there's a large IP minefield associated with the technology. And it definitely impacts especially industrial applications where to own the thing that you make is critical. So part of that is going to be important for the solution that we're going to offer, and I'll talk about that in just a moment. Okay, so Inscripta wants to be able to enable this upper quadrant here, all right? So that's really what we're all about, okay? So how does it work? How are we going to get there? So if you look at today's technology, if you wanted to do CRISPR-based editing, and you wanted to do sort of the non-trivial editing where you're not just knocking something out, you want to actually replace it with a defined sequence, then you can work in, say, a liquid handling format where you have partitions with individual isolates that you want to create. And then to carry out the CRISPR process, you need the guide RNA to direct where the double-stranded break is going to be. And importantly, the homology arm that's going to repair that specific break, not some other, needs to be paired with it. That process can be carried out and is carried out now today, but it's immediately obvious that this is not a scalable process. It's very hard to get to tens of thousands or more of individual isolates using this format. You can work in the low dozens to hundreds, or if you're a really big, say, biofoundry, and you have the infrastructure, you can get to thousands with a lot of, with a pretty heavy lift. So instead of treating the well as the partition, what if we had the cell as the partition, right? So imagine a world where we could take the critical components of the CRISPR editing technology and deposit them into the individual cells, okay? So in particular, what you need to be able to do is have the guide RNA and the homology arm covalently linked together, along with, say, a barcode to enable facile trackability, which I'll talk a little bit more about shortly, and to deposit that into the cell, right? So if you had, say, a Maxwell's daemon that could direct at the molecular level where certain entities went, you'd be good to go. But we don't have a Maxwell's daemon, but we have an Andrew Garst. So Andrew Garst is one of the co-founders of Inscripta, and he had this brilliant insight to say, okay, I need to get this material in a defined way into individual cells. So let's do that by covalently synthesizing these sequences and use array-based oligonucleotide processes that are highly scalable. You can make thousands, tens of thousands or more of these defined sequences on chips. And now you can take that material off the chip and then clone it into your expression cassettes and then transform it into, in this case, it's a picture of microorganisms being edited. That, along with a key component of the system that I want to share with you, I alluded to on the last slide, is a proprietary nuclease that was discovered at Inscripta and carries out very efficiently the editing process, much like the other nucleases that are out there, like the Cas9s and the CPF1s. But it's our own nuclease. It was independently identified and developed, and we offer it with very favorable licensing terms to the worldwide research community. And in particular, you fully own the rights to your inventions and your discoveries. You make an edit, it's yours to keep, okay? Without any reach-throughs, royalties, or licensing terms. So this, as we've talked to customers and we've gone out to the world, has been incredibly enabling for a lot of applications that were sort of out of reach for people who couldn't navigate the licensing issues. Okay, so this technology was reported on first in late 2016 in Nature Biotech by Andrew Garst, the lead author, and others. And was followed on by a number of papers from leading labs, including folks like George Church and others, sort of demonstrating the scalability of the process has been well confirmed. But something that's not widely appreciated is that the actual editing rates in these systems can be quite low without a lot of advancements associated with the editing process. And that's something that Inscripta's worked very hard on, and we've essentially been able to solve, is the editing rate criterion for the platform. Okay, so after you have that tube of edited cells, what do you do with it? So these are two sort of primary workflows that you can think about for phenotyping your system. One is to take those tube of edited cells and then subject them to a pooled experiment where you're letting the cells kind of fight it out against each other under some selective pressure. Then you would sequence the populations with that barcode that I talked about to look at which of the editing cassettes are enriched or depleted in the population. And I'm going to show an example of what that looks like here in a minute. Alternatively, you can array those individual isolates from the pool on, say, microtiter plates, and then do things like microfermentations. This is a very typical workflow for industrial biotech labs that usually result in some high throughput direct detection at the end of the day to get the phenotype. But whether you go each route, at the end of the day, you have genotype and phenotype information that you combine that you use to decide what are the next set of experiments that you want to do. Okay, so that was a lot of setup. So now we're actually going to get into some data. So let me show you some examples of real live editing experiments where we made large libraries, and then we've sampled from that population to identify what the edits were that we put into the system, to identify and verify the edits that we put in. So this is an example of, so this is all in E. coli. So what we're doing here is we're putting in a promoter along with a ribosome binding site in front of every gene in E. coli. That'll be a primary edit type I'll show you some more examples of here in a minute. But this is just one isolate we pulled out showing a 61 base pair insert went in and it was clean, a couple sequencing errors there, but it indeed carried the large non-trivial edit, the kind of edit that we think is important to really open up genome engineering. So that's nominally an expression change, right? Another kind of thing you could do is, say, do a triple stop at a location in a gene to sort of knock it out. And here we're doing a 9 base pair swap to put in this triple stop, likely leading to a loss of function for that gene. And then another kind of swap is just a codon swap that is shown here, but it's a library of different codons. And the reason you do this is because you want to be able to do that saturation mutagenesis that I talked about earlier. So here we're representing lots of different amino acid variation at one position in a protein that we wanted to target. And then it's very straightforward to run this across the whole protein to generate all the single amino acid changes that you want, individual changes. Okay, so those are some of the variety of edit types that actually we're going to use in a demonstration application. And the demonstration application that we chose was for lysine biosynthesis. So we, Inscripta, are not a metabolic engineering company in the sense that we don't want to make products. We don't want to make small molecule products or proteins. We want to create platforms that will let the world carry this out themselves. So we wanted to pick an application that would sort of light the fire or the imagination of our customers in the community, larger community, for what's possible with this technology. So lysine is a pretty profitable molecule that's out there that is used as a feed additive. and we think demonstrates a lot of the concepts associated with pathway engineering. So what we did is we said, okay, here's the pathway in E. coli for lysine. And so let's identify, or we identified the 19 enzymes in the pathway to lysine along with a couple of ancillary enzymes, and then subject that to various forms of mutagenesis at the amino acid level that I kind of showed on the last slide. So that represented a major source of diversity. And that in and of itself is kind of unprecedented, right? All that history of enzyme engineering that I've talked about that's quite successful, you'd engineer one, maybe 2 or 3 enzymes. Here we're doing 19 parallel enzyme engineering efforts at the same time. But that's not enough. We want to go outside the pathway because there's so much we don't understand about genomes. Let's go ahead and take a broad, fully agnostic approach to generating diversity through various means. One is a genome-wide knockout, where we try to knock the gene out, every gene in E. coli, in 2 different ways, with stops at different positions. And then also, let's actually insert promoters in front of every gene to try to regulate expression. And not just one promoter, let's do a ladder of promoters. Okay, let's have high strength, medium strength, and low strength promoters, because you don't always know which direction you need to go. So that represents sort of the variety of edit types that we were interested in making. And in the end, we designed and ran through the editing process 200,000 edits. So this is really an unprecedented number in terms of any kind of biological engineering that I think anyone's ever sort of been a part of, certainly for me. And so this represents a wonderful capability that allows us to interrogate in a heretofore unthinkable scale, the entire genome. So with 200,000 edits in hand, what do you do with all that potential diversity? So I'm going to show you 2 different ways that we phenotype this library. One is through a screen. It was that lower path where we array individual isolates. We do microfermentations, and then we do a direct detection to figure out how much lysine the individual variants have that carry edits. And what we're showing here is on the x-axis the number of edits in each of these library categories. And up at the top, it's shown the KOs that I talked about, the pathway amino acid mutations, as well as that promoter ladder that we talked about earlier. And on the y-axis is the fold improvement over a base strain of E. coli. Turns out it was MG1655, produces just a little bit of lysine. We wanted to start with no assumptions and see what we could do with just a wild type strain. So we're looking at the log2 fold improvement of any mutant we happen to pull out of that population over the control. So you can see in our primary screening, we were able to achieve some interesting possible hits that had more lysine that could be used in further evolution. We identified numerous primary hits in our screening, but like with most screening campaigns, you have to then turn to verifying them in a retest. So this is actually the retest data. I'm going to walk you through this a little bit busy slide. So first I'm going to start with a key enzyme in the pathway called DAPA, which is well known to be a key control point in the flux towards lysine and is actually allosterically feedback regulated by lysine itself. So it represents an interesting known target and allows us to see whether we can recapitulate some of the known discoveries that have been reported in the literature. So for DAPA, what we're looking at here is the retest fold improvement performance in 2 different media conditions. And what you see is there's a number of amino acid mutations that we pulled out of the screening and were significantly improved in one or both conditions. And these in circles are all individual amino acid mutations at different positions that conferred significant improvements. And in particular, that allosteric site that I mentioned, we were able to recapitulate quite easily through our screening process. So that sort of validates the approach and the technology to some extent. In addition to that, we showed that through the screening campaign, putting a high-strength promoter in front of DAPA was able to confer a huge improvement in performance. In fact, a 1,000-fold improvement all by itself in lysine titer. It was actually our biggest hit in our initial screening campaign. So that's DAPA. But what I want to show you, in addition to that, is sort of genome, sort of library-wide, which includes both pathway and the rest of the genome. How do the hits sort of turn out? So what you see as you sort of run your eye over the results is that there's now lots of genes that showed up as hits in the results, as well as a variety of edit types. The circles are amino acids, you have frame shifts, promoters, and stops, and you can see that there's a wide variety of edit types represented and a large number of genes here that are targets of interest. And what's really interesting is that in addition to a number of other pathway enzymes that showed up as important, we also identified 17 new targets outside of the lysine pathway that confer some significant boost in performance of lysine. And this is really interesting because a lot of the genes are uncharacterized or unknown. There's this Y-ome in E. coli where like a third of the genes have no known function. And so they wouldn't show up high on the list of sort of rational targets to intervene on. And yet this sort of broad a-rational approach and capability to generate lots of diversity turned out to produce a lot of useful genetic material. Okay, so that's the screening part. But screening, as powerful as our infrastructure was, we could only sample about 10% of that very large population. So what we wanted to do was try to develop a phenotyping platform that could evaluate more of these edits. And so a good colleague of mine, Dan Held, who's in the audience here today, set up these really nice selection experiments where he looked at challenging the system that is producing lysine with a toxic inhibitor that is pretty much like lysine but for the sulfur that when present, this molecule wreaks havoc with the metabolism of E. coli. And what you're asking when you present this anti-metabolite to the system, you're asking the nature to come up with solutions that overproduce lysine. That's one escape mode that we're really interested in finding. So with that setup, what Dan did is he ran several biological replicates where he treated it and also had the control condition without the anti-metabolite. And then he ran that for some number of hours. And what you can even see is there's kind of partial clearing in the treated cultivations. And so there's interesting cells in there likely that are surviving this anti-metabolite and hopefully producing more lysine. Those entire collections of cells we can read out as a pool using that barcode technology that I talked about where you just sequence the plasmid. And you can get really nice data on what's enriched and what's depleted. And so that's what I'm actually going to show here on the next slide. So what we're looking at here is count data from those cultivations that was on the last slide. And for the control condition as well as the treatment condition, you're looking at all the different pairwise combinations of biological replicates. And the numbers represent the count statistic, the log 10 count statistic for an individual cassette, an editing cassette in the population. And so the takeaway is that if it was a high count in one rep, it was a high count in the second and third rep as well. Okay? So very nice biological reproducibility in the control as well as the treatment condition. So that establishes your baseline for any experiment like this. But then what you're really interested in is if there's differences, you want to try to understand which of those editing cassettes are enriched or depleted. So in particular, we're looking for things that are enriched. They have a higher count in the treatment condition. They've survived that selection pressure relative to their counts in the control condition. And so if we double click on that cloud of data there, and again start with just DAP-A to not overwhelm you with the data, what we're looking at here is the depletion or the enrichment of various cassettes in your system. So over here we're looking at things that have a high count in the control and are depleted in the treatment. And so these are things that are likely disrupting function of the DAP-A enzyme. So these are all amino acid mutations, and then also a low strength promoter in front of DAP-A reduces lysine flux, okay? Which is not surprising at all. But what we're really interested in are these candidates over here that are a combination of both amino acid mutations that have high counts in the treatment relative to the control, as well as are stronger promoters. So this recapitulates what we saw in the screening, that overexpression of DAP-A leads to a benefit, a survival benefit in this setup. Okay, so that's DAP-A, but now just like we did in the screening, let's look at all the rest of the edits as much as we can in the system. So what we're looking at here is, on this Circos plot, is the counts in the control condition, that untreated control condition where the radial direction is the counts for the different edit types that we have. So the black ones are the amino acid changes, the blue ones are the genome-wide knockouts, and then the dark greens are the genome-wide promoters. And this gives you a nice visual sort of feel for our ability to target the entire genome. So if we just start with the amino acid changes and pull out what's the single best amino acid change that's statistically significant in one of our enzyme, our pathway targets, we find a couple of heavy hitters. 3A and DAP-A again are very important. But interestingly enough, all the other enzymes in the pathway had some, if modest, but significant, statistically significant impact on lysine, or survivability in the lysine selections, I should say. So that's interesting. That's within the pathway. But what does it look genome-wide? Well, it turns out the genome-wide knockout library produced another 10 hits all throughout the genome. And one in particular was also found in the screening. This is a lysine transporter, but it's also involved in regulation. So it's not just blocking the anti-metabolite. It really is doing something to increase, flux towards lysine. So that's great. We'll take that diversity, the genome-wide knockout diversity. We'll take another 10 hits. But what's really interesting, or perhaps even more interesting, is the genome-wide promoter ladder. So this is really a kind of a novel intervention genome-wide that was heretofore impossible without with this technology. So this represents upregulation and downregulation all across the genome. And we hit upon another 90 or so promoters of varying strength that conferred benefit to lysine or anti-metabolite insensitivity. Okay, so 100 new targets outside the lysine pathway were discovered. And this is just briefly sort of the corroboration between what we pulled out of the screen. This is the rank order of the things we pulled out of the screen. This is the best to the lower among the hits. And then we looked for every instance where we found that exact same editing cassette in the selection, and then just simply looked at the correlation between the two different modes of phenotyping. And so what we find is that the things that are highly ranked in one phenotyping system are generally highly ranked in the other. So this gives us confidence that a lot of the things we're pulling out of the selection are actually probably truly better for lysine production. Okay, so there's all kinds of interesting things you can do with this data. So one thing we wanted to do was to ask, are the different edit types driving biology in different ways? So in particular, does the KO drive things differently than the promoter, as you'd kind of expect? And here are a few examples that represent different directions. So on the y-axis, we're looking at the log2 fold improvement standardized by the standard error of the statistic that shows that for the KO, we're getting enrichment for these genes in that treatment condition, whereas a promoter, the higher strength promoters, are depleted in the population. And then here it's kind of a reversal of fortunes where these KOs got depleted in the treatment condition, but these promoters got enriched, okay? So that's nice. That kind of confirms your basic understanding that loss of function and overexpression should be doing different things. So one could ask, well, this might just be cherry-picked to prove a point. And so we want to look at, for all of the genes in the system, does this sort of profile, what does it look like? But it's hard to look at thousands of genes in Excel bar charts. So instead, let's take this high-dimensional representation and then map it onto a lower dimensional subspace using principal component analysis. And so what we're looking at here is the clustering of the different edit types in principal component space. So in particular, we're looking at the knockouts here. These two black spheres represent the two different ways you can make knockouts. They pretty much had the same effect, across the whole, genome. Which isn't surprising at all. Interestingly enough, the knockouts are completely separated in the first principal component from the promoters, as you might expect, but it's nice to see the confirmation. So as you separate the KOs from the promoters, which are plotted here, you find sort of structure within the promoter space as well. So the high-strength promoters are kind of off in their own area of the PCA space. The low-strength promoter is down here in its own area, and then the medium-strength promoters are sort of somewhere in between, okay? So importantly, this shows our ability to do knockouts, upregulation, and downregulation of expression all in the same experiment. Okay, so I talked a lot in the beginning about not just generating diversity, but then starting to stack together mutations to get improved function. And this is sort of an early look at what that looks like in our hands. So what we did here is we said, let's take one of our best backbones or backgrounds to then do further optimization in. And it was that one DAP-A variant that had an important allosteric change. And then let's put a library on top of it, right? And start to improve it further. And so what you're looking at is just a sampling of the high-strength promoter library on top of that single DAP-A mutant. And we're seeing things that are now 1,500-fold improved compared to the 400 or so that we started with after that single mutation. So that's for some random promoters, all of which we've checked and are different in the system. But one promoter in particular, probably not surprising, if you take this 1,000-fold improved high-strength promoter in front of DAP-A and you combine it with this 400- or 500-fold improved amino acid mutation in front of DAP-A, you end up with something that's now 10,000-fold improved over your starting strain, okay? With just those two mutations. All right? So sort of putting it all back together, to go back to our sort of strategy slide, this is the version of pathway and genome engineering that we want to enable. We want to be able to carry out massively parallel single edits, as we've talked about a lot here today, but we also want to be able to create multiplex combinatorial editing workflows. So this allows you to start recombining rapidly the beneficial material that you've discovered in the first round. So that's where we're headed. And so just an early look at how this might start to look is we're now demonstrating the ability in both E. coli and yeast to be able to introduce multiple edits per cell. So here what you're looking at is for the different organisms, the fraction of cells with certain numbers of edits. So most cells have one or more edits, or isolates I should say, and the distribution is 2, 3, up to 4 edits per cell in the case of yeast. We'll drive this further and higher over the course of time and as we continue to develop the platform, but this is already getting us into a region of combinatorial complexity that we can now start to navigate using some of the methods that I talked about earlier. Okay, so in summary, where are we at? So hopefully I've been able to persuade you that two key aspects that have been well developed in the protein world over a number of decades, diversity generation and combinatorial optimization, can result in very powerful biological optimization strategies. But that's not enough. You also need the tools to do this at the genome writing scale. And for that, you do need that unprecedented scale, high variety, large numbers of genomic loci. You need superior performance in terms of high edit rates. And you need that greater access. We want to make this easy for people to do, and we want you to own everything that you create. So with that in place, we believe that the dawn of rapid genome engineering is here. So this will allow you to create and test many more ideas, discover and exploit new biology, and importantly, as I've already mentioned before, focus your expertise and resources on your own questions and goals, okay? And so, final slide, Inscripta. So who are we, where are we at, what are we trying to do? So we are developing the world's first scalable platform for benchtop digital genome engineering, okay? And what that means is that we're currently in early access with this platform, all right? And the technology will be commercialized this year for E. coli and yeast, and we will enable both single edit and combinatorial editing workflows. And this is important, and I think is worth thinking about if folks are interested in this platform, is it will consist of software, instrument, and reagents. And the instrument in particular will have a desktop or benchtop format that will allow you to run the editing process in a push-button walk-away format. So you would load your cells and reagents, hit go, and 2 to 3 days later, depending on the organism, you would retrieve your edited cells and then carry out your experiments with that material. All right. We are going to launch this whole platform at SynBio Beta coming up in early October, and so look for a lot more information at that time about the rest of the details. Thank you very much.